Now, I'm pleased to present Dr. Dress. You can come on up. She's a professor of neurology, the director of the Lois and Solia ALS Clinic, the Les Turner ALS Center at Northwestern Medicine, director, MDA Clinic, Northwestern University, Feinberg School of Medicine, a couple more things. She has her medical degree from the Medical School of Tunisia. Then completed her neurology residency at the University of Illinois at Chicago and a neuromuscular fellowship at Northwestern. She's board certified in neurology and neuromuscular medicine, has been treating patients with ALS in the Lois and Solia ALS Clinic for nearly 20 years. She also leads the Les Turner ALS Center's ALS clinical trial program, and the scientific advisory board is a scientific advisory board member for NEALS. With that, welcome, Senda.
Thank you. Good afternoon. It is really my honor to introduce the keynote speaker for this year, Dr. Angela Genge. Now, you can read her biography in the booklet you have, but I just took a few snippets. So Dr. Genge is the executive director of the ALS Center of Excellence at the Montreal Neurological Institute at McGill University, Canada. For about 20 years, she ran the clinical research unit, and under her leadership, it grew to become one of the largest clinical research, neurology clinical research centers in the country. And Dr. Genge is recognized as a leader in the field of ALS and clinical trial design. She's really recognized internationally. And her exceptional care and dedication to her patient and clinical research had earned her the prestigious Forbes Norris Award in 2018 and many other distinguished awards. Her exceptional care and also her expertise in drug development is really invaluable. An October day, about 3 years ago, a day that is forever etched in my memory, we were on a Zoom call with Biogen and the Valor or the Tofersen study investigator to discuss the top-line result. We were all stunned, and we could not believe the negative results. There was a very kind of long silence, and we were all very disappointed until Dr. Genge came up and said, well, you guys should remember that the field of MS only took off when they had an MRI as their biomarker. So perhaps neurofilament is our new biomarker, and a drop that important should be taken into account even in the face of negative clinical result. Perhaps it takes longer for function to recover. And she was right. Patient needed more than 6 months to see an effect. Now, many of you may not know, but I started my North American journey in Montreal. I did not meet Dr. Angela Genge there. However, she took me under her wing about 6 years ago, and I love to think of her as my clinical research fairy godmother. She always had the best advice, from always pack a swimsuit to how to best run your clinical research program. She is the forever optimist. You never see her without a big smile on her face. She is a champion and a fierce advocate for her patient, determined to make this disease, ALS, a treatable one. So please join me to give Dr. Angela Genge a warm welcome.
Thank you, Senda. The other part of that was I blamed it all on the statisticians. I think I actually blamed them out loud on that call, but that's a whole other story. It's been a great day and a great visit, and it's an honor to be here, Senda. So thank you, Hendy. I've actually known about Les Turner for a very long time, and I was friendly with previous leaders at Northwestern and at Les Turner, so I'm absolutely delighted to be here. I'm going to take you on a journey, and the answer's not there yet, except for Tofersen, but I really do believe tofersen is priming the pump for some really important breakthroughs where we're going to have disease-modifying therapy for a lot of different patients with ALS. We just have to be smarter. Pietro spoke of some really important new developments that are going to lead to some really, really cool new drugs. And I spoke a bit about this on Friday. Today, I'm really going to talk about what we do with clinical trial design and when do we stop thinking a 20-year-old trial design is good enough. And I guess I'm, pushing the envelope and suggesting there's more work to be done, but we are so close to serious improvements in ALS trial design. This is my disclosures. And first, I'm going to spend a minute on the FDA and the EMA. They're really not such bad people. We think they're bad people, but they're really not such bad people. Their plea to us, and I've heard it from the FDA and from the EMA, is figure some things out so that when we look at the results of a clinical trial in ALS, we have more to work with. And what are they asking for? They're asking for things as different as biomarkers that can truly predict clinical outcomes. So we have it in SOD1 with neurofilament, at least once we've had it. They're asking for things like, when you do the ALS FRS, don't impute to 0 every time you lose a patient because you're doing yourself a disservice. I'll mention that again in a minute. But they're also asking for us to really get more interesting and novel trial designs and approaches to the results so we can demonstrate a change if there is one. Because some of the things we've been doing for the last 2 decades are hiding positive findings. Of course, we can't really do it unless we get really good at drug development, and that's what we're doing with tofersen, and there are things they're following on. So my message about the regulators are they can only work with what we give them. We sometimes disagree with how they interpret what we give them, and we really don't like the CAFs. And the CAFs is a combined measure of function and survival that really sets the bar very high. But it's our responsibility in clinical trials and drug development to do something better so they can work with something else. So I'm going to run through some of our major categories and give you an idea of where we're going with some of these. And if you've ever been involved in a clinical trial, or if you run a clinical trial, a lot of this will be familiar to you. Imagine this is a list. We mix these up, put them in a blender, spit them out, and we're still doing the same trials where we're doing, we have to do safety and tolerability, but then we're looking at this functional rating scale developed in 1998 by a movement disorder specialist, not even an ALS specialist at the time, and survival. That's what we're doing. So what I would propose to all of you is let's get better at all of this, including how we do the ALS FRS and survival, but not just those two. Let's get some other things in the game so we can see a response before we have a change in survival. So this little diagram is one of my favorites. I'm going to take you around this diagram. I think each of these have important messages. And you see I put target engagement without anything underneath. When Pietro figures out how to measure cryptic exons reliably, then we know we have a home run. So I'm leaving that to you. But in the meantime, there are other things we can do. So I'm going to start with the most obvious. Probably everyone that's ever walked into an ALS clinic knows about the ALS FRS. What people don't realize is that the original ALS FRS was designed in 1998 and validated. Then the R, which is respiration, was tagged on, but never validated. And then we developed different ways of training people to use it in clinical trials. And what we did was we developed it in silos. So it is validated, at least without the R. It's accepted by the FDA and EMA and Health Canada and Australia and Japan. And we believe that there's a 25% change is significant. This is actually came from a survey of experts in the field. So when we talk to ALS patients, much less than 25% may be considered significant if you're a person living with ALS. But we know that there are challenges, and a lot of those challenges came out of the tofersen trial. So one trial has contributed a lot to our understanding. So it's a questionnaire. What could go wrong with the questionnaire, right? Well, I can name several things that have gone wrong. Still, the most important thing we can do is improve this, because if we can reduce the variability in the scoring from this questionnaire, we have a lot of ways to compare the ALS-FRS scores with previous trials and with longitudinal studies. But let me just run through a couple of the things, not everything on this slide. You realize there's no clock up here, Cinda, so I could be going till 4 o'clock. Somebody's gonna flag me down. Oh, you're the clock. Okay, Telly's the clock. The ALS-FRSR, until 2 years ago, was trained differently by the NEALS Consortium and by the TRICALS Consortium. The difference was so much that a rater doing the assessments could have a difference of 4 points on the same day, same assessment, because of the differences in the training and the way the questions were used. On top of that, the Japanese have their own. They go back, they use the certification developed in 1998, and they've never evolved it. So you run an international trial like tofersen, where you need every patient counts in the world, and you are now dealing with 3 different ways of training and scores that can vary by at least 4, just with the same rater, then you got an issue, right? So I'm super happy for a lot of reasons that neurofilament got us over the finish line, and that the effect of tofersen is great enough to overcome this. But we've started on a campaign, and I'll call it a campaign, not like your campaign that's ending tomorrow. It's a little bit more civilized, not much. But it's a campaign to improve the way we use ALS-FRSR in clinical trials until we have a better score. Because yeah, we can throw it out, but we can't wait for 5 more years until we have a new score to do another drug trial, right? So we've started a campaign. There are a couple of different elements of the campaign. One element, this is the 4 levels of functioning, and this is what it looks like. One element was that the group for NEALS and the group for TRICALS was training, their questions were being administered differently. So we got everyone together, we got the key people together, and we harmonized. So they each had to give a level, and I'm telling you that's not such an easy task. They each had to give a level, and I showed up late, so they had to do it before I arrived, to come to a harmonized version. And lo and behold, they did. What they did was line up their questions. So they were asking the same questions to get the same score. And their interpretations of the answers, they lined up. And so we have a harmonized European North American version. But that doesn't include the Japanese. So our next step with the harmonization is to meet next month with the Japanese to bring them on board, so that you can now, at the end of this process, be able to run a trial internationally and know that the training and execution of this questionnaire is the same whether you're doing it in Japanese or Dutch or English or Spanish, so that everything is consistent. We also discovered something else. We discovered that at the same site, different raters talking to the same patient on the same day can come up with different scores. Not as dramatic as the difference in the different ways it was being trained, but still enough that if you're looking for small changes, it's going to be lost. So now we've made a couple of big advisories. In a clinical trial, the rater has to follow the patient. So the same rater for the same patient throughout the duration of the trial. So you can have 2 or 3 or 4 raters, thank you. But they have to stick with the patients. And the way that got uncovered was COVID. You guys all lived it, we lived it. There were sites in the Healy platform trial in which every person on site quit during COVID. So they had to restart and retrain every single individual. Not to name names, but one site went down to the only person left standing besides the neurologist was the secretary. And the secretary was doing the ALS FRSR. It caused me to cringe. And so we said, okay, so is there another way to do this? There is another way. The other way is self-report ALS FRSR. Now, traditionally, the attitude has been, oh, you can't give this to the patient to do by themselves. You have to have a certified rater, or else you're going to get the results are going to be uninterpretable. Well, it turns out that there are at least 4 different self-report versions of the ALS FRSR. There's an American one. There is a one from the Netherlands. There's a one from England and a one from Germany. They're not the same, but each of them within their databases have recognized that patients are actually more consistent than raters. And you wonder, well, that doesn't make sense, but it does, because if a patient is an optimist, they're always going to score themselves better than someone else. They don't change over time. If they're a pessimist, they'll do the same thing. So what doesn't change is the patient. The person reporting the results actually is pretty consistent in their attitude towards how they're doing. And so there's a whole group now that's looking at recommending self-report ALS-FRS to be used in clinical trials in ALS, to really improve our use of it, get rid of more of the variability so that we're not missing important signals because of a functional outcome measure that is unstable, that's bouncing around. Things do bounce around, but we want to get it more consistent. We know that the ALS-FRS really predicts survival in the speed with which it changes. And so we know we need to harness this rather than throw it out. Lots of people like to throw out the baby with the bathwater, but we have so much data on the ALS-FRS. We're really trying to reform it, bring the prodigal son home rather than just throw it out and start again with something else. The data, just for your knowledge, that really pushed a couple of us to look at this came out of this trial. So you can imagine there are 2 or 3 things that affect the tofersen development. One probably was that it takes a few weeks for the antisense oligo to start to have an effect. And we see the effect in the biomarkers, which I'll talk about in a minute. But to get the clinical effect to show up, you probably need closer to 6 months after the effect begins. And with tofersen, that would have been 9 months of a placebo-controlled trial. If we'd gone to 9 months, we may have gotten it approved 2 years earlier. So we had to do a lot of work to sort of get the full data set and demonstrate this benefit. The ALS-FRS was part of that problem, and it was really working with the group at Biogen that were struggling with the results to uncover some of this variability that really hurt us in this trial. The other thing that we think is really important in our new and improved ALS-FRS world is the statistical strategies. So you think, well, it's an outcome measure. We've been told how to analyze it. There must only be one way. Let's do it. Well, it turns out one of my colleagues in the Netherlands put a PhD student on it. So PhD students, if you show up one day, you might get a project like this accidentally. I don't think he volunteered. And he put the PhD student on looking for the different statistical methods used to evaluate the ALS-FRS. He looked at 8,700 studies, even excluding duplicates, excluding other aspects that would make it hard to compare. You go right down to the trials. You still have 38 different ways of analyzing the ALS-FRSR. So how do you compare one study with another study, right? So this is another campaign. So once I got the guys to write a paper and harmonize, we're almost there with Japan, and we started the project on the self-report. This is the third part of the sort of ALS-FRSR campaign that was initiated. It's to get recommendations from the world of statisticians. Like I said, I blame them for almost everything. Not everything, but almost everything. To give us recommendations, which 2 or 3 SAPs should we use in different phases of studies? How do we implement it? I have to say the FDA has taken this up with a vengeance, because they're frustrated that we're throwing so many different things at them. And so they're really encouraging, they're part of the group, and we have a group, international group, including some Healy people, some Tricals people, some Germans, as I call them, and several leading industry people who've worked on ALS clinical trials for a long time, to come up with a list of recommendations. And the white paper I am aiming for the end of this year, so it's not very far away. We're looking at things like, how do you manage missing data? When a patient with ALS dies, they're actually not at 0 in the ALS-FRS usually. They're somewhere at 20, 22, 25. So how do we improve the way we do patients disappearing, dropping out of a clinical trial? In a variety of ways, we need to come up with a better number. How do we work with the different assumptions that we create? How do we get better than the CAFs in terms of our primary outcome measure in the registration trial? So I don't have the answer other than making friends with statisticians, but the group led by Ruben Van Eyck and Eric's Macklem, they're really going to put some time and effort to help us get better at our analysis of clinical trials. And hopefully we'll never again have a situation where the statistical analysis plan really created the problem we had when tofersen was read out. What else can we do? Well, you know, the 20-year-old trial design is a measure of function and survival, but we are way beyond function and survival now. And I'm going to advocate for much more robust use of biomarkers in ALS clinical trials. What are biomarkers? Well, biomarkers can be a number of things. They don't all have to be read out of biospecimens, but that's the handiest way because it's easy to draw blood. You can draw it frequently. You know, we've had the benefit of having a lot of cerebrospinal fluid in the intrathecal technologies, but blood is good, right, as a biomarker. So working on different kinds of biomarkers is really important. You've already heard about neurofilaments at least once today. And although we do not believe that every single drug that has an effect in ALS must move neurofilament by 30, 40, 50%, we do believe now that if a new drug moves neurofilament, lowers neurofilament by a significant amount, it appears to need to be more than 20%. It looks like 40 to 50% is enough, that we believe that will translate into a positive effect in a clinical trial. So how else can we use neurofilament? Well, it can be prognostic, so we can use it as an inclusion criteria, and so you only choose patients who have elevated neurofilament in a clinical trial. Or we can use it to identify pre-symptomatic patients. That's what we're doing in ATLAS. We're following their neurofilament until it starts to go up. And in the ATLAS trial, people who carry SOD1 mutations but are asymptomatic, living normal lives, are being followed every 6 months with a neurofilament serum test. And once their neurofilaments start to go up, they're followed more closely with the idea that if you treat really, really early, you can actually have a profound effect on the course of the disease. I mentioned controlling for disease heterogeneity. If it's an elevated NFL level, you can predict a more rapid course. And the final way we're looking at neurofilament is the tofersen way, which is if you have a treatment response, neurofilament will go down. Now, it's not simple, guys. Nothing is ever simple in ALS trial design. There will always be issues around these markers, but at least we have one that can be used to stratify, to prognosticate, and to be a covariant to help us in our analyses. Are there other biomarkers? Well, I am a huge proponent of target engagement biomarkers. I just want people in their labs to get better at measuring some of the targets, because I don't do that. I ask others to do it. We know that when they approved tofersen, it was on the totality of the evidence, which included the fact that tofersen knocked down total SOD1. So they had a disease-modifying biomarker, and they had a target engagement biomarker. So now we're into some really funky, groovy treatments for sporadic ALS. The antisense oligos are now going after different targets for patients who don't carry a gene mutation. I can name at least 4 that are either in clinic, about to go in clinic, or are in a pipeline to be in clinic in the next year. Each of them have a different target. Every one of them has a different target. So the current one in clinic is Stathman. The one just about to go in clinic is calpain-2. The 2 following up, one is targeting lipid metabolism, and the other is targeting another protein that we see a SNP elevated, UNC13, in genome-wide sequencing of ALS patients. Each of these have ways for us to turn the targets into measurable biomarkers. They're not easy, but that really is the next step in development. One of the things we have found is that the cryptic proteins that were mentioned this morning are very, very hard to measure. They don't seem to be around for very long. So we're actively making our biomarker development on targeting the restoration of the full-length protein that, or in the loss of function, or the knockdown of the full-length protein or mRNA. But stay tuned. I think this is going to help accelerate drug development in ALS. This is the SOD1 example, and you can see it went down really nicely. And then the patients, this is delayed start, early start. So the blue are the patients who were in placebo, and the green are the patients who started from tofersen at the get-go. And you can see it actually is really impressive that they were able to knock down total SOD1 and keep it down. Are there other ways that we can talk about biomarkers? Senda pointed out that I have this big MS envy thing going on. They did not do, our MS colleagues did squat to find MRIs. Just telling you, that was a gift given to the field by the very talented radiologists and physicists. In the 80s, they created the MRI. I still remember looking at these UBOs, unidentified bright objects. And then, you know, the penny dropped quite quickly that this was a pattern that was seen in the MS patients who were diagnosed by something, this'll really date me, double dose delayed contrast CTs. That's how we used to diagnose MS before MRI came along. And the first at least 4 drugs that were approved for MS were approved based on the biomarker, the change in the MRI. They did not have a clinical benefit by the time the drugs were approved. So we know that things other than biological specimens can give us a biomarker, and the one that I think has renewed legs is electrophysiology. And what we're doing is we're going forward and backward. EPhys gives us a readout in particular of the distal motor axon and the neuromuscular junction. We've tried a few different things over the years. The traditional Muni technique, you had to be a board-certified senior electrophysiologist to be able to do it reproducibly. The more relatively new Munex technique had the patients flipping from side to side, backwards and forwards. And so it was, although providing stable outputs, it was just physically demanding on the patient. So where are we moving now? Well, we know about that hyperexcitability is a really important feature in ALS, and that hyperexcitability actually also tracks survival. We know that from a couple of studies, particularly retigabine, that even something as simple as CMAP can show a trend to significance by preserving CMAP. In the treated group and the placebo group, the CMAP dropped just the way we always have known. It correlated with some other electrophysiologic techniques, particularly strength, duration, time constant, and the Rio base. And although retigabine has not moved forward, mostly because it turned people blue, or at least their eyes blue, so it was taken off the market because we didn't want Smurfs invading the ALS clinic, I guess. But seriously, there is a lot of work now being done on drugs that are also being developed for epilepsy, on hyperexcitability and that preserving function. But at the end of the day, because this is a biomarker talk, what I am proposing and what a number of people are working on is using both CMAP longitudinally in early phase clinical trials and a technique called MScan, which is a way of using CMAPs to build out a more robust measurement indicating stimulus response. So we're going from Muni and Munix into MScan and back to CMAP, but we actually think this is worthwhile and may actually give us some non-clinical readout to support target engagement. Why do I have strength in here? Don't we have to do strength in ALS clinical trials? Why would we not do strength? Because it's such a directly related to the disease course. Well, strength testing has taken on a lot of different forms over the years. It has demonstrated in Valor to respond with the patients who were on the drug from the beginning. They actually had a difference in their HHD megascore, handheld dynamometry megascore. But why wouldn't I just be all on the bandwagon for strength? Well, I guess I'm going to, again, sort of upset Dr. Scheffner, because I'm going against his long-standing philosophy. It doesn't seem to bother me much, because I keep doing it. Strength testing for a patient visit in a clinical trial takes 45 minutes out of their assessments. They are exhausted at the end of a robust strength testing. The strength testing measurements you get have a ceiling and floor effect. If you change the rater and you go from a big physio male who sort of works out on his spare time to a petite little physio, you actually can change the scores just because the person doing the evaluation has a different height, a different strength, etc. But the biggest reason I'm suggesting we have to rethink this is there is not one drug in 20 years that's been approved on the basis of changes or improvements in strength. So although we may have to keep it in some form, I'm really advocating for a change in approach in strength testing in ALS clinical trials. I'd like to spend that 45 minutes on a clinic visit on something else. And there are other things that are less exhausting that may be actually equally robust. Time to event outcomes is another piece where that falls in. I'm going to potentially move that, talk about that a little bit more. But time to event outcomes are ways to take any one of these things that I've talked to thus far, and including pulmonary functions and using a change in the score as your outcome measure as opposed to survival sake. Time to event outcomes are borrowed from oncology, but in oncology, they measure the size of a tumor. We don't have a tumor to measure, but we do have certain events that are predictable and therefore can be identified and allow patients who are on placebo to go into the treatment arm, particularly in like a phase 1b/2 trial. And an example of what we can use in ALS for a time to event outcome is a time for a strength in a limb to drop by a certain percentage, a time for the ALS-FRS score to drop by a certain number, or the time for the pulmonary functions to drop by a certain percentage. All of these would give us new opportunities. It's not quite a biomarker, but it is an outcome measure that it can make our trial designs, evolve them into more adaptive designs. One of the things that I think we forget about, and I never like to forget about the patient. One thing I think we should take home from the VALOR program is just how good the patient-reported outcomes were at predicting clinical outcomes. So, of course, I have stories. So the story around this one is, and the statisticians at Biogen even agreed with me. They didn't know they were agreeing with me, but they agreed with me. The changes, the improvements seen and the changes when you compare placebo to early start tofersen in the ALSQ5, in the EQ5D, and in the fatigability score, all showed early differences, statistically significant actually at certain earlier, not as early as neurofilament or total SOD1, but earlier than the ALS-FRS-R. How does that make sense? Well, just think about it. Think about it. The patient's starting to feel better, and not in a placebo response kind of way, but they're starting to feel that they're moving better. But they can't actually document a change in a score on the ALS-FRS-R, but they're seeing that they're walking upstairs a little faster. They're moving, they're using their utensils a little better. In fact, as we were following these patients, and we had a lot of patients in the program, we saw a difference in these patients before we could record the difference. And the ones who started tofersen earlier in their disease course definitely did better. But we had a control group, which I think is the thing that's made me really high on these PROs and really pushing to have them higher up in our statistical analysis plan in terms of prioritization. We had during COVID, we were open for business, and we had a significant number of patients in the SOD1 VALOR trial and in the two C9orf72 trials, WAVE and Biogen. And we'd have multiple patients on in the same day from the different trials. And what was very clear was the difference in their PROs. We didn't know what they were on, but we could tell that patients coming in on tofersen were just reporting changes. Even if their ALS-FRS-R wasn't changing yet, they were consistently reporting changes and improvements in their PROs. PROs are patient-reported outcome measures. They're all questionnaires. And the patients in the C9orf72 trials, which were ultimately negative, never had that finding in their visits. So it wasn't, because it was between trials, it didn't unblind one or the other. But I think it really reinforced for me, again, that we should be listening to the patient voice more in clinical trials. So my aim is to improve our use of the patients themselves and to improve our use of science through biological biomarkers to make more robust clinical trials. Never forget pulmonary functions. You have amazing pulmonary medicine specialists in your clinic. You're probably leading the way in some of these things. I'm sure you are. I think we forget how powerful PFTs are in ALS and how important and how objective the readings are. And at some point, we need to advocate for the pulmonary function testing to become a more significant outcome measure in terms of our statistical analysis plans. I'm not a huge fan of remote pulmonary function, mostly because my first 2 or 3 experiences, it was difficult for the patients to learn to use the technology. So the technology is an issue. But it does allow for more frequent testing. But again, I think it's really an important thing we don't look at very often. And we did see the benefit of pulmonary functions in the VALOR study. Almost finally, I'm going to talk about survival. So the definition of survival is a bit of an issue. Currently, our definition of survival is survival without non-invasive ventilation. Or survival to death, right? So one of the issues that has been uncovered most recently in a discussion, and we're going to have it again in Montreal at the ALS MND, is that non-invasive ventilation is not uniformly used around the world. There are very huge regional differences, and even in the US, there's a huge regional differences. So it is an artificial survival measure because access to non-invasive ventilation is an issue. So currently, the way we try to read it is when is non-invasive ventilation of benefit? Like, at what point do we decide to offer non-invasive ventilation? So is there a way for us to standardize, even if access is an issue, but standardize the way we assess for the use of non-invasive ventilation? I think that's possible. We certainly will have trouble standardizing access to non-invasive ventilation. You go from, not everybody, every country is heavily involved in clinical trials, but if you go around the US, I think you would find that different regions have way more access than others, and different regions assess earlier than others the need for non-invasive ventilation. This particular issue also is an issue. Survival is an issue because there are one or two other points around this. One, those with really rapidly progressive disease, so they're actually short survivors, are underrepresented in clinical trial populations because they never make it into clinical trials. They're the runaway trains. We saw it a lot in the SOD1 population, where some of the mutations that we really wanted in the trial, we never got the patients in because they deteriorated so quickly. We couldn't even get 3 readouts of their SVC to remain stable enough to actually screen them into the trial. The other issues we have with the survival time, so NIV is one, rapidly progressives aren't in the trial population, so you lose that data. Survival is an issue in shorter trials, which are early phase trials. And now, to make it even more challenging, a fairly substantial part of the trial world, the world in where most of the ALS clinical trials are performed, have medical aid in dying. So you suddenly lose survival as an outcome because they're choosing medical aid in dying, even if it's very close to their survival, to when they would go on to non-invasive ventilation, is still far enough away that that you can really handicap your analyses. It was a big issue in the cytokinetics trial, and their interim was negative. So their interim analysis for futility was met, but they had already recognized that medical aid in dying was causing a problem. So it's not it's not a problem, it's just something we have to start to account for so that we don't make bad decisions on outcomes of good drugs because we have these new variables that are affecting our old outcome measures. So another issue to think about when we measure survival. The other problem with survival is it's really, really important for the EMA, and the FDA is comfortable with function. So we've got this weighting issue between the two major regulators. One, the EMA, that really wants to see survival data, and two, the FDA, that if you see a benefit in function, they accept the arguments that it would lead to a benefit in survival, and we haven't yet gotten the two together. So we haven't really solved survival in the ALS clinical trials. So it just heightens the bar that we have to meet to get a drug, to demonstrate to everyone the drug should be approved. I'm going to spend my last few minutes on different strategies that we're trying to use to improve the speed with which we can do clinical trials. And the biggest one and the most scrutinized one right now are platform trials. So there's different ways of doing platform trials. They really came out of oncology. There is one very famous platform program in Alzheimer's, and our most famous one is obviously the Healy platform trial. The benefits of platform trials are you testing multiple drugs at the same time, and you're sharing your placebo. The challenges of platform trials is the lack of control you have over what arm the patient goes in, or some aspects of the trial, because once the platform trial has started, it's a continuous trial. Now, the Healy has done a great job of going through a whole bunch of different molecules to date. It's now undergoing a major amendment in recognition of a lot of what we've learned. So one thing we believe now is that a phase 2 trial in ALS should be 9 months long, placebo-controlled. And that's really based on a couple of different things, but one of them, again, is the tofersen trial, where we didn't really see maximum clinical benefit until 9 months after the trial started. There's also evidence that it takes that long for certain biomarkers to change. And so with respect to how important it is to not end a trial too soon, the Healy is actually doing that along with a couple of other major amendments. But it really is an important new way of approaching new drugs in ALS. It doesn't really adapt to novel drugs as well, like intrathecal, but it definitely has the benefit of sharing the placebo arm. The current readouts are going to be for the integrated stress response inhibitors. So it's a drug from Denali and a drug from Calico, now with AbbVie, that is a very important new target that may have a particularly good effect in C9orf72 patients. And we'll wait to get that readout and see who comes into the trial next, which molecules come next. But it is a really important development in ALS clinical trial design. There are other forms of platform trials. And the other forms can be earlier, they can be phase 3. The next configuration, I guess, of a novel trial design is going to come with the master protocol that the TRICALS is putting out, which is a different way of doing a platform style, but it is really focused on phase 3 or registrational trials. So the Healy is really a phase 2 trial design. The master protocol that TRICALS is going to implement is really a phase 3 trial design. So lots and lots of different things going on. What are my take-home messages? There are a few. Biomarkers have to be integrated into every single program now. at least at the 1B2A period. And the biomarkers that are identified as good reflections of drug activity, either because the drug, the biomarker is a target engagement biomarker, or whether it is a disease-based biomarker like neurofilament. We really need to be able to see those movements early in order to really push forward into a big phase 3 program. There are people who believe, me being one of them, that some of these biomarkers we may need to implement as inclusion criteria in our registration trials. Because one of the big criticisms of the last 4 failed trials is that they took all comers. So they made it too hard to actually show a benefit, because they took a very heterogeneous group of ALS patients and put them in the same protocol. SOD1 was the perfect experiment, because you had to have a mutation. And in fact, you had to have one of a small group of mutations in the SOD1 gene. The other side of the coin is what we've done in the last 4 sporadic trials, in which we took absolutely everybody, whether they had elevated NFL or not. We took absolutely everybody with the diagnosis of ALS and above a certain SVC cutoff. And that may be a mistake. So we'll see. We're looking at this in a number of trials that are currently somewhere in phase 2 to see if we can actually make tighter inclusion criteria and really aim these drugs at patients who have a chance of responding. So I think that's where the biomarker story is coming. The critical analysis of trial design, I said it's necessary. I think I've strongly proposed that we look at more robust phase 2 trials so that we can streamline our phase 3 trials. So let's start looking more and more at seamless design, where patients roll from 1 to 2 or 2 to 3. Let's make sure we add in a variety of outcome measures to see which ones move in conjunction with our target engagement and our clinical. So once we get to phase 3, we can choose a patient population that's most likely to respond to the target that we're treating. The better use of the primary and secondary outcome measures really fall into number 2, because I think we have not done our fair share in giving the regulators the right things to work with. Let's look more closely at how we're analyzing function and survival, and maybe we should be doing function and neurofilament as co-primary and not worry about survival. Survival will come. Maybe we increase our focus on PROs. So if you've got function and PRO changes in the same direction, maybe that's stronger than function alone, the way we measure it. Open label extension is a must. It's the most important thing that we have really changed in the last few years, in that every program needs to have an open label extension. It's not just because a patient has contributed to the main study, so they should get ethically access to the drug. The open label extension gives you really important long-term biomarker and other readouts that can change your outcome, your final analysis from a negative to a positive. And so the best example we have of that recently is the value of the open label extension to the tofersen program. That was really the other part of the, what did they call it? The totality of data that they saw that led to the approval of this drug. Because what we saw in clinic was the longer you treated them with tofersen, the better they did. If they were started early enough, they recovered slowly over a period of time. It wasn't something that happened in 6 months. So I think that that is a given. And then finally, and I'm saying this one more time in case you missed it, is adaptive designs that save us time, make us more efficient, skip the downtime between phase 1, phase 2, phase 3, and really have a more adaptive, seamless program really should be our new standard. And we learn with every part, and there are things we will drop in our registration portion of the trial, but we really should be making the most of every person's time commitment to these individual clinical trials. And with that, I think I'm good. This is my team, and thank you for listening. And you can see from the picture, everybody speaks minimum of 2, maybe 3 or 4 languages. So we're covered.
Excellent. So questions, please.
You can always count on me for a question. I want to applaud everything Angela's done. I've known her for many, many years, so full marks. Couple comments. One is, if you'll recall in the Riluzole trial, it actually was scheduled for 18 months. So trying to do 6-month trials or even 9-month trials has been problematic from the get-go. And even though in the States we terminated that trial after 15 months, the original plan was for 18 months. That belies the problem that I think we all have, both from the clinical side and from the research side, and that is, we need better therapies, man. This trying to struggle through changes in the ALS FRS or ALS FRS-R or one of those things. Boy, if we had something more potent, you wouldn't have to worry about whether you're doing a 9-month trial or a 12-month trial. I mean, there are lots of other things we could say about that, but, you know, haven't got there yet.
Thanks, Bob. So I won't tell you that I was too young to be in the Riluzole trial. I agree with you, but that was kind of my Friday message. It is super duper exciting right now in how we're applying new technologies in ALS. I think we're going to change the game just by the use of new technologies to get to other parts of the pathways, whether it's Stathman or UNC13 or calpain-2 or a pathway that is amenable to siRNA therapy. And then eventually we're going to be able to shuttle everything via new shuttle systems from IV use. So I think we are on that. But what I don't want to have to do again is spend an extra 2 years getting a drug approval that clearly worked because we used some traditional trial design issues. So we've got to do both. Find the new drugs and improve the way we do the trials. Robert? No, but it's your birthday.
Oh, well, I'll be very fast. Okay, so what a wonderful, wonderful whirlwind of a talk. So thank you so much. And I think that you're right. We are sort of like packed full of pulmonologists that do ALS around here. So we're sort of spoiled. That being said, I think that all of us, if I'm speaking for our chest group, would say that NIV is not just a Band-Aid in this disease, that it's disease modifying. I think there's enough data to support that. So I'm going to throw 2 questions at you relating to that. Number 1 is, should oxygen and CO2 be considered biomarkers, just like every other biomarker? And overnight oxygen CO2 studies could easily be done and added to the biomarker panel. And then the second is, I understand the access to NIV problem probably better than anyone here. My nurse will tell you the number of times my cell phone goes off because some program 6 states away can't figure out how to order the NIV. That being said, given that it is bioactive, why are we not controlling for it as a disease-modifying agent as part of these trials? In other words, we track who gets Riluzole, but we know that the impact of NIV is 3 to 5 times what the impact of Riluzole is. And NIV use and compliance doesn't even show up on your case report form. So I guess my question is, what do we need to do to change that?
So I actually think it's a completely legitimate question. I actually think that we need to actually consider additionally using FVC or SVC as a key secondary. But the NIV is a really important issue. We don't manage this well. We're not acknowledging the difference in start times. We're not acknowledging the effect on lifespan. There are a bunch of different things we don't acknowledge. So we're just starting. I scratched the itch on the ALS FRSR, and we're just pushing strength. And then this other issue, which is how we manage the pulmonary status, I think is the next thing that we want to tackle. So you may be invited to help.
Sign me up. And anybody from the Les Turner Foundation who's here, listen about how important it would be if we could develop a fellowship for pulmonologists that want to go out there in the world and do this. Just saying.
Thanks, Lisa. Okay, birthday boy.
So I just wanted to thank you so much for providing the depth of the overview of really how difficult it is to do clinical research, even with something that appears so straightforward. Just people get weak.
So that, I wasn't trying to show that how difficult. I was just trying to show how much we can change.
Fair enough. Going from the macro, just one little micro question I had is, why can't we do actigraphy? It's like a wrist measure of movement, put it on the limb that's weak but not paralyzed, and just integrate that over time. That seems like numerical and time-based. Why can't we do that?
It's actually a great question, because there is some work on this, Robert. There are people who have used the equivalents of smartwatches. We're just a little bit early. We haven't totally sorted out, but we're coming at it from 3 different directions. And here is where I think some of the muscular dystrophies have actually started behind us and have already passed us, okay? I also, between like midnight and 2 a.m., work on FSHD and myotonic dystrophy, and a colleague works on adult Duchenne projects. And in those spaces, there are 2 or 3 things that are starting to get into ALS now. One is the EMERALD system. Some of you must know about the EMERALD system. It's relatively new to me, but it's a box that you put in a corner of a room. It was, the first publications were on sleep studies, because it records sleep, but it records activity as well. And it's now being tried in one program as a non-wearable, but like an environmental measure of activity. Because there is a correlation between movement during sleep and degree of weakness in ALS patients, and I think that's the main measure there. The other one, which is in a few of the FSHD programs, is Microsoft Connect. And I find this fascinating because they've been, the drugs don't work yet, but that's besides the point, we'll get there. They've been able to measure movement on just, a television, big television monitor, using the Connect programs. And they've actually turned it into an objective outcome measure in FSHD, and a fairly robust one. So yes, actigraphy, there has been a home program. It's not yet worked out how to use the smartwatches, but I think there's really 3 categories. There's that, there's the Emerald type devices, and then there's these gaming systems that are actually really robust, and just we need to figure out how to convert them. But I agree with you that would completely replace strength testing, because you'll have objective movement. Good job.
I just want to add to that, the voice recording also are being used
Yes.
through oral analytic, the apps to record speech and use that as a framework. I want to go back to the study duration. And it's really hard for a phase 2 study that is more than 6 months. I think, is it worth it really to extend to 9 months, taking into account the dropout of patients that with disease? should we focus on biomarker for phase 2 and have a shorter study and then have a longer study for phase 3?
So I think you've hit the nail on the head. One of the problems we're having is we're conflating trials. We're doing a 1B and trying to call it a 2A, and we're doing a phase 2 proof of concept, but then trying to get approval. So a true phase 2 doesn't need to be more than 6 months if you have robust biomarker and outcome measure data coming out of it, because it is not a registration trial. The problem we have when we do a 6-month phase 2 and call it a phase 2 is often the sponsors, if they get even the slightest signal, try to turn that into some sort of an approval. And that part is on the regulators. They've got to hold firm, and the sponsors don't like anyone holding firm. So the in-between there is to start with a proof of concept phase 2. If you see a signal, then you make the amendment and take it to a registration trial. But don't try to use 6-month phase 2 data to get your final approval, because we'll keep getting the situation where things are approved in North America, they're not approved in Europe, everyone's complaining, but complaining of 2 different things. So we do need to clean this up. And I've seen the same thing in 1B to 2A, where people are really designing a 1B trial, but then they're so anxious to get further, they'll then halfway through decide it's a 2A, throw in a few extra patients and call it a 2A. So we've got to actually hold to the original principles, but make the trials themselves more robust. You know, I'm telling people we should be doing motor point biopsies in some of these programs because our targets are at the neuromuscular junction. Why don't we, in the early phases, do some muscle biopsies? I like doing muscle biopsies, it's a problem. But, you know, if you look at Clotilde's work, we should be able to do motor point biopsies and actually document if we're hitting our target. You know, think about that, but we're not quite there yet. I haven't convinced enough people to do that or do MRI, so. I have about 30 seconds, so go for it, Andrea.
I'll make it really quick. So my question is, and I agree with you, I think we have seen problems in the past with cohorts in clinical trials being so heterogeneous that we're not seeing the signal we need to see, and you raise the issue of being more homogeneous. Makes perfect sense. How do you sort of answer the need of the patient community today to sort of maybe create some balance there to give people more opportunity? Is it an expansion of EAPs, or what do we do or can we do anything to create more balance?
So I think the balance is created by understanding the individual patient's disease better and direct them to the right trial. So there's an assumption that every drug should work for every patient, and yet we know that's not true. So, and we have a lot of drugs in development. So the more we understand about the individual patient, so we're going to start doing UNC13 SNPs on every patient to make sure in those trials the right patients go in. Should we not be doing that in the big Miracals trial? It's an IL-2 trial, but not everyone responded. So are we just missing the boat by not identifying those that are most likely to respond? Because if Joe, who has an inflammatory driven ALS, wants to go into a program that's really focusing on a particular protein, he's choosing the wrong trial for himself. So I think I would advocate for a slightly different approach and sort of match the patient to the right program.
So
you're the very last, because Telly's going to get upset with me.
I know, but we have some questions in the chat, so I want to
okay.
honor them as well. They say, why can't we use tofersen for sporadic ALS patients? I think it would be important for them to hear from you. Why can't tofersen be used for sporadic ALS patients?
So it goes right to Andrea's question. So in theory, if we were to identify patients without a known SOD1 mutation, who we can confidently say their disease is driven by SOD1 pathology, and there's some evidence out there that there's a percentage of sporadics that are not necessarily TDP43, that is few percent are, then tofersen is appropriate. But it's not appropriate for a sporadic patient whose disease is being driven by TDP43 pathology. We're treating the wrong protein. We are treating the wrong protein. So that's the main reason.
Wonderful. So there's one more question. What strategy and inclusion criteria do you recommend for sporadic ALS patients to make the population more homogeneous when we don't have any biomarkers?
Well, so I think we have biomarkers that we don't necessarily use to the full extent in our inclusion criteria. So I think what I'm really advocating is the inclusion of more biomarkers, whether they're very simple or much more complicated, in our, not necessarily early, because it doesn't, in safety and tolerability trials, it's not as important, but certainly in our registration trials, we should be ensuring that just like when you do a breast cancer trial, you do all those markers to see what they're positive and negative in. We should be thinking that way in ALS. I think that's where we're going. That's what personalized medicine is about.
Yes.
But we do have more biomarkers than we recognize. We acknowledge. We recognize them, but we don't acknowledge them, and we need to figure out how to input them into these trials.
Wonderful. Maybe one last question. Neurofilament is actually related to axonal degeneration. But ALS is very heterogeneous, and not all patients display axonal degeneration. And how if it is mitochondria or ER or other aspects of the disease, then would neurofilament be a good biomarker for them or not?
So that's a really good question. When we use the term heterogeneity to mean a lot of different things in ALS, right? But fundamentally, at the end of the day, there's axonal loss. And that's where the neurofilament comes, whether it's ER stress proximally in the cell body or a pro-inflammatory state at some time during the course of the disease. The challenge we have is that we need to determine what makes a more homogeneous population. Because it'll always look heterogeneous. Is it really that heterogeneous? Only our biology will tell us.